So I'm Joshua Pajak, and I suppose there are enough Europeans present to say my name is Joshua Pajak. And I'll be explaining the work that I recently finished up in the ARIA lab at Duke University. And I'm wearing a red sock shirt because I recently, as of this past Monday, started a postdoc position in the Kelch lab in Massachusetts. And so I felt it was very fitting. And so this is a second talk, kind of follow up to a talk that Mark Murray gave in a flash talk, The Lord of the Broken Rings Part 1 yesterday. And so I need to give kind of a brief background, kind of rehashing and recapping a lot of what Mark covered. Just because there were a lot of wonderful talks, I was flipping back and forth between a lot of these talks. And so I want to make sure that we're all on the same page going forward. So we study for this project Phi-29 assembly, which if PJ tells me the truth, everyone here loves Phi-29 assembly. And so I'm sure everyone's kind of already familiar with this. But the idea is that these viruses form this prohead that's completely empty to begin with. And then they use this powerful molecular motor, which is an ATPase, to translocate DNA to near crystalline density inside of the virus. And so some landmark results that I think would be remiss without talking about come from single molecule experiments. And so these single molecule experiments have shown that the motor doesn't just package DNA continuously, but rather it packages DNA in a biphasic dwell burst model. And so essentially during this long dwell phase, the motor is exchanging spent ADP for new ATP, the catalyzed hydrolysis. And during this dwell phase, the motor is not translocating DNA actively. And then during this burst phase, the motor hydrolyzes all of the bound ATP and uses that energy in order to translocate DNA, roughly one helical turn into the viral capsid. And so through kind of this tour de force of single molecule studies, we have this understanding that ATPase activity is sequential and ordinal around the ring. ADP exchange for ATP, for new ATP is also sequential and ordinal. We also have some kind of confusing results. And so those confusing results when I started were, it's a 5-member motor, but we only have 4 translocation steps, but there seems to be 5 hydrolysis. And so one of those hydrolysis events is not necessarily providing work to translocate DNA, but it's likely doing some coordinating function. And so that was a fairly confusing result from the single molecule experiments. And so this model is kind of, I feel like where the state of the art has been for the last several years. But the problem with it is that it doesn't really tell me how the motor actually works. It gives me this really nice set of states that the motor transitions to, and there are even kinetics associated with how the motor transitions between those states. But it doesn't tell me what the motor looks like at any one of these given states. It doesn't tell me how the motor, what the conformational changes are to transition between those states. And it doesn't tell me what molecular mechanisms actually regulate the activities to ensure that ATPase activity is ordinal and sequential and all of these very fine-tuned processes. It doesn't really give me any insight on that. And so I teamed up with some wonderful collaborators. And so just to kind of, again, talk about what Mark talked about, we recently solved a CryoEM reconstruction of the Phi-29 packaging ATPase installed during packaging. And we find, for instance, the main result from this was that the ATPase GP16, the N-terminal domains, which is the ATPase catalytic core, forms this helical pitch as each subunit tracks around DNA like a corkscrew, ensuring that each subunit is engaged with the phosphate backbone in a nearly identical way. And then the second result coming out of this was this crystallography result, which shows that in the absence of DNA, the motor from a homologous motor prefers to be in this kind of planar conformation. And so right away, we started fiddling around with this, trying to say, well, we have what we think could potentially be 2 end states where the N-terminal domains form this helical pitch, and potentially another end state where the N-terminal domains form this planar pitch. Could we just infer from the Berkeley kinetic model what the steps could potentially be to go between these 2? And so we found that once we posed the question like that, the steps kind of wrote themselves. in the sense that in order to transition from this helical pitch to this planar pitch, there are only 4 transformations of subunits aligning from a helical to a planar interface that need to be accomplished. And so right away, that gives us an insight as to why there might be 4 translocation substeps from a 5-member motor. And so we propose that there are all of these intermediate states based on this kind of Berkeley kinetic model that are getting us between these 2 end states that we feel like we determined experimentally. But now the question really is, this still doesn't really tell me how the motor works. It doesn't tell me how potentially these events are coordinated so that they happen sequentially and orderly around the motor. It doesn't tell me what the driving force for any of these conformational changes are, et cetera. And so I think that's really one of the advantages that molecular simulations can provide, is giving us an inherently dynamic way to interrogate dynamic processes. And so I'll just kind of highlight a bunch of the results. If I went through every single result, I think I'd be here for another 20 minutes. And so I feel it's better to not do that. And so I feel like key results for us to understand is how the motor is predicted from molecular simulations to change its conformation based on nucleotide occupancy. So over here on the left-hand side, we see the apostate simulation. And in the apostate, we have this lid subdomain color-coded in yellow, which mediates most inter-subunit interactions. And in the apostate, the lid subdomain is predicted to be flexible and extends away from the catalytic core of the motor. In the ATP-bound state, our simulations predict now that the lid subdomain rotates over the catalytic core, so ATP binding and also ADP binding induce this lid subdomain rotation. And so just immediately from kind of observing that, and we have the understanding that the lid subdomain mediates most inter-subunit interactions, we felt that maybe ATP binding, ADP binding is a way for 2 subunits to change the way that they interact with each other, perhaps to modulate between the planar and helical configurations. The next result that we got from simulating the entire pentameric complex is we can now start to really interrogate dynamic transpromoted interactions. And so for instance, here we can directly predict how ADP unbinding might occur. So we see the ADP molecule initially bound to the P loop, the critical lysine of the cis-acting subunit. And now we have this trans-acting arginine, which was originally proposed to be kind of the arginine finger, but now with some updated data, we're starting to suspect that this arginine that PJ talked about wonderfully yesterday perhaps is not necessarily a catalytic moiety, but is rather sensing and helping exchange ADP. And so now our simulations are starting to predict how that might be. When the ADP dissociates from the cis-acting subunit, it associates to this trans-acting subunit. So really putting together, the end goal of this project was to kind of expand upon this helical to planar model, which is supported by some single molecule results, and try to figure out what's going on that's being obscured by this arrow. And so putting together results about how the lid subdomain works, about how the motor is predicted to change its grip on DNA based on nucleotide occupancy, and based on how the trans-acting interactions occur, whether it's planar or helical, what we currently think happens is that 2 subunits are initially in this helical configuration, and they're driven to that helical configuration because they're both binding DNA very tightly. Hydrolysis at the top subunit releases that DNA grip, thus releasing the constraint for this helical configuration. So hydrolysis at the top subunit now allows the lid subdomain of the neighboring subunit to undergo this rotation that's predicted, driving DNA into the capsid as the 2 subunits align to a planar configuration. Then ordinal hydrolysis activity is induced because trans-acting residues that catalyze hydrolysis are really only properly positioned in this planar interface, and they're not properly positioned in this helically offset interface. And likewise, we can take information about how ADP might potentially be released from a binding pocket. When the subunit no longer binds ADP, when it passes through this APO state, we understand kind of this lid subdomain flexibility. And so we think what happens during the dual phase is initially you have two subunits which are in this planar state. ADP release is catalyzed with this trans-acting arginine residue, and that helps remove ADP. When the subunit passes through this transient APO state, its lid subdomain is predicted to be flexible, which we attribute that flexibility for the ability for the subunit to track back down and realign with the helical phosphate backbone of DNA. And once it aligns, now new ATP binding causes tight DNA gripping, locking it in that helical configuration. And so the last part of the talk I'd like to talk about that really is testing kind of the first prediction is that the basis of our model is not really anymore that ATP hydrolysis and phosphate release causes the force generating conformational change, but rather phosphate release after ATP hydrolysis really only causes a small conformational change in DNA gripping that allows a larger ATP binding driven conformational change to occur. And so we were wondering if we could use molecular simulations to investigate that. And another way of posing that question, can molecular simulations tell us some conformation that's unique about the ADP bound state versus the ATP bound state that could support this hypothesis? And so for sake of time, I'm just going to very quickly rush through the background, but there is this very nice experimental result predicted of how the active site can modulate between these two positions and the active and inactive pose. And so we were wondering if perhaps the enzyme modulates between those two positions based on whether hydrolysis has occurred yet or not. And so I did some free energy calculations, and I showed that for all of the viral packaging ATPases that we studied, in the ATP bound configuration pre-hydrolysis, the motor prefers one conformation of the active site. And post-hydrolysis in the ADP bound, that conformation switches from this right-handed side of the free energy landscape to this left-handed side, which is kind of this inactive pose. So now we're seeing that the molecular simulations are predicting the way that the motor modulates its conformation based on whether hydrolysis has occurred yet or not. And so now the question is, how does that information in the active site get transmitted to DNA gripping residues? And so we predicted this slight conformational change in this alpha helix, which flanks the active site, based on whether or not this active site conformational change has occurred. And so for all of the simulations, we predicted that there's this conformational change in the alpha helix. And so we really hoped that there would be some experimental validation for this. And so when we proposed this alpha helical change, we assumed that from the simulations that the ADP bound and the APO should look very similar to each other. And so when we actually looked at some solved crystal structures, we saw that indeed in this solved crystal structure, at least a partially ADP occupied subunit looks very much like an APO subunit and distinct from the ATP bound subunit in that regard. And so we think that there's this slight conformation change in this alpha helix that's driving the propensity for DNA gripping or not. And so with the last minute and a half, I just want to mention some really, I think, kind of nifty techniques. And so with a lot of molecular simulations, I think the power is you're generating a lot of data all at once. And with that data, you can use certain machine learning techniques. You can project that data into latent space and use autoencoders. Or in this case, what we use is a fairly simple technique, mutual information. And so over the simulation time, we're generating configurations of our protein, which are dictated by this kind of angle configuration space, the backbone Ramachandran angle, and then certain rotamers. And so we look at the probability over the course of our simulation of observing two residues in the same two residues in a certain configuration, weighted by the probability of observing those independently from each other. And so if they occur a lot together, we say that that has high mutual information, and we infer that those residues are communicating with each other. And so we can calculate mutual information allosteric signaling pathways that directly connect the ATPase active site to DNA gripping residues and all of the viral packaging ATPases that we studied. And we can mutate that signaling pathway, which in particular, this glutamate 58, running out of time. So I'll just say when we mutate that glutamate 58, computationally, it seems like it would abrogate DNA gripping. And then also experimentally, the glutamate 58 mutation also abrogates DNA gripping while maintaining ATPase activity. So we really think that is the signaling pathway. And so with that, I'd like to give the acknowledgements for the NIH for funding, Exceed and Anton for computational time, Gaurav Arya, my old PI, and then the wonderful collaborators, PJ, Mark, and Brian, and then all of the Arya lab. So thank you. I'll answer questions in the chat afterwards. I think we should move on.